Hi everyone, welcome back to my channel. And guys, we were doing this concept of Human Genome Project. So in the previous videos, we have completed about till the methodology, that how we have started all this, right, and how we are proceeding with this project. Now there in this lecture, we'll be covering about some salient features of Human Genome Project, that what this genome project is about. So the human genome consists 3164.7 million base pairs. Okay, this is the number which has been found out after the research has taken place. Okay, ji. So human genome, it has 3164.7 million base pairs. Correct. The average gene which is present consists of 3000 bases. वैसे तो यार बहुत सारी genes हैं, लेकिन we are talking on an average. On an average, a gene consists of around 3000 bases, but sizes vary greatly. The largest human gene being dystrophin, 2.4 million base. Okay, this is an average, but the largest one is around 2.4 million bases. The total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 1,40,000 genes. Whereas the total number of genes which are present in our human genome accounts to around 30,000. The functions are known. Okay, almost all 99.9% nucleotide bases are exactly the same in all humans. You know, there is only 0.1% variation, due to which we all look different. Otherwise, 99.99% we are same. Functions are unknown for over 50% of the discovered genes. We do not know only about 50% of discovered genes. We do not know about their functions. Less than 2% of the genome codes for protein. Less than 2% codes for proteins. This line is also important because this is only carrying some relevant information. Repeated sequences make up very large portion of human genome. They are continuously repeated. Repetitive sequences like 1, like 2, like 3, they are stretches of DNA sequences which are repeated many times, sometimes hundreds, so sometimes thousands. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Now, if you talk about these repetitive structures and if you think that they might also code for some information, so that is absolutely not true. They do not code for any relevant hereditary information. They are just for this process of chromosomal structure, dynamics, and evolution. Chromosome 1 has most genes and Y has the fewest. So chromosome 1 has 2968 genes, whereas chromosome Y has 231 genes only. Correct? So this is the number of genes present on that chromosome. Scientists have identified about 1.4 million locations, different locations, where single base DNA differences are there. That is single nucleotide polymorphism, SNPs, which occur in humans. Now, what are they speaking over here? Single base DNA differences. This information promises to revolutionize the processes of finding chromosomal locations of disease-associated sequences and tracing down the human history. Because these SNPs are only related with single nucleotides, so they help us to find out various locations where diseased cases can be found out. These were some features which were relevantly, discovered from this Human Genome Project. Then there were certain applications and future challenges. Deriving meaningful knowledge from DNA sequences, this will define the research through coming decades. This enormous task will require expertise and creativity of tens of thousands of scientists. One of the greatest impact of having the human genome sequence may be enabling a radically new approach to biological research. If we discover something new, that will really lead to a new track. Past में researchers have studied one or few genes at a time ताकि हम उससे relevant information derive कर सकें. While the whole genome sequences, the new high throughout technologies we can approach systematically and on a much broader scale. अगर हम सब करना चाहते थे तो हमें एक advanced technology का wait करना पड़ा. They can study all the genes in a genome. For example, all the transcripts in a particular tissue or organ or a tumor, or how tens of thousands of genes and proteins work together in interconnected networks define the chemistry of life. Okay, to how we know about a particular tissue or organ, to how know about thousands of genes and proteins working together, we need to find out how the chemistry of life is organized. So with this, we finish up and wind up with Human Genome Project. The next topic in the next video, we would be starting with DNA fingerprinting. So thank you guys for watching my lecture, and that's all.